Good afternoon and welcome to this month's Dean's Research Seminar. Before we begin, I'd like to acknowledge the traditional owners of the land I am on today, the Kulin Nation, as well as the traditional owners of the land you're situated on. And I acknowledge the importance of Indigenous knowledge in our academy and acknowledge that Indigenous Australians have been custodians of the lands and waterways of Australia for thousands of years, and I pay my respects to their elders, past and present. Professor Amir Haji Noor Mohammadi from Melbourne Veterinary School will present today's Dean's Research Seminar on strain identification of veterinary pathogens, the benefits of this and the challenges of this. Let me tell you a little bit about Amir. He completed his Doctor of Veterinary Medicine at Tehran University in Iran in 1990 and then worked as a lecturer in public health in the School of Veterinary Science at Urmia University in Iran. He then completed a PhD here at Melbourne University in 1998 and was a research fellow after that at the Walter and Eliza Hall Institute of Medical Research, studying vaccines for leishmaniasis. He returned to the University of Melbourne in 1999 as a research fellow and has made his way through the academic ranks to become the professor he is today. He's made important contributions to the faculty, which have included being director of the Werribee campus and for the last 10 years, head of veterinary anatomic pathology, head of avian medicine, and Deputy Director of the Asia Pacific Centre for Animal Health. Amir is the coordinator of the DVM3 subject, poultry and avian birds, and also teaches in a number of different areas in the DVM. He's authored over 120 peer-reviewed journal articles, 15 books and book chapters, is an associate editor for the Avian Pathology Journal, and is sought internationally as a speaker. He currently supervises 4 RHD students and has previously completed another 19. He was awarded the Victorian Poultry Industry Person of the Year and the prestigious International Houghton Trust Lecture Award in the area of avian diseases. He is an avian disease diagnostician and has been trained and interested in combining conventional diagnostic techniques such as gross microscopic pathology with more advanced molecular techniques. His research has primarily focused on pathogenesis of infectious diseases of poultry, especially those caused by mycoplasmas, herpes viruses, adenoviruses, coronaviruses, and chlamydia. Please welcome today's speaker, Professor Amir Haji Noor Mohammadi. Amir, over to you.
Thank you, John. Thank you for the opportunity to present some of the stuff I've been busy with for the last 10 or 15 years. And also thank you for pronouncing my surname properly. So well done, John. At the outset, I need to acknowledge that I'm on the lands of Woiwurrung people, which is Werribee, in fact, who have been custodians of this land for thousands of years, and I acknowledge and pay my respects to the elders past and present. Before I start, I need to warn you, this is not one of those presentations that sort of talks about specific projects. This is basically just giving you an overview of what one of the things, in fact, has kept me busy for the last 15 years. And this is actually strain identification of veterinary pathogens, predominantly poultry pathogens, in fact. So with that, I'm going to, in fact, share my screen. Here we go. Can you see my screen properly?
Yes, perfect, Amir.
Beautiful. So I might just put that away. Okay. All right. So the first slide, I thought I'd just give you a little bit of idea about what we are as a group, Asia Pacific Centre for Animal Health. So basically a part of the greater microbiology group. Some of us are on teaching and research positions, and obviously we do our own fair share of teaching, but there are also a lot of people in the group which are basically recruited with the grant money that comes from different entities. One of the major, in fact, objectives of the group is to train PhD students or master's students, and also some of the other activities, especially we do at Werribee, includes some fee-for-service activities in terms of diagnostic service we provide to the various sectors, but also these obviously diagnostic activities contribute to our teaching and also to some of the research projects that we run within the group as well. So in terms of diagnostic activities, in fact, we have sort of the whole package, including conventional pathology, clinical microbiology, thanks to Mark Miranda, serology, and also molecular tests. And today, in fact, I want to talk about specifically molecular tests and specifically about strain identifications that we do within the group. So here is a list of strain ID tests that we do within the group. And as you can see, there's a long list of tests that we offer to the poultry industry or other industries. The one that I've highlighted here are the ones that we have also strain identification tests for them as well. So it's not just, let's say, detection of pathogens, but it also includes identification of the strains involved. I like to, in fact, use ILT, infectious laryngotracheitis, as a model to demonstrate to you what benefits we get out of the this strain identification within the group for the good of the, in fact, faculty and even greater community. So, and I'd like to take you through the journey I've had with this particular disease. And in fact, a lot of us within the group have been working on this particular disease, including Joe Devlin, Glenn Browning, and Andreas Diaz, and some other people, Alistair Lejeune as well. And just to tell you what it took us, in fact, to develop tests for this particular disease and especially strain identification techniques, and what benefits this particular test has had for the faculty, and also the challenges we had to face and tackle throughout the years as well. So for those of you who haven't heard about, in fact, ILT, it's an infectious disease caused by a herpes virus, or alpha herpes virus to be accurate. And it actually causes upper respiratory infection in commercial chickens. And so in terms of clinical signs, the severe form of the disease, in fact, causes very severe dyspnea and open mouth breathing and gasping. And sometimes, as you can see in this image there, also causes conjunctivitis. So it's a pretty nasty disease, quite often involved in mortalities in the commercial poultry as well. And at the moment, it's arguably the most important and significant disease of poultry in terms of economic losses it induces. So on top of the obviously economical aspects of the disease, it's a obviously very significant welfare concern for the obviously birds as well. The gross pathology of the disease includes tracheal or laryngeal lesions, and could be sometimes hemorrhages or sometimes casus plugs, like what you can see, in fact, in this image here. And in fact, to the point that sometimes these casus lesions, in fact, block the passage of the trachea, and the birds, in fact, are unable to breathe properly. And that's why they actually have open mouth breathing and gasping. If you do histopathology, you'll see obviously very severe necrotizing tracheitis and sloughed epithelial cells and fibrin and all of those, which actually you see grossly as casus exudates. And typically you'll find some intranuclear inclusion bodies within the sloughed epithelial cells. And if you do obviously further work, you realize the herpes virus sitting there. And it's a disease which has been, in fact, causing problem for the Australian poultry industry for a very long time. And I'll tell you a little bit more about the history of our group, in fact, working with this disease in the next few slides. Here you go. All right, I took this image, in fact, in 2004. As you can see, back then we had few outbreaks in the layer industry in Australia, and based on that, we applied for a grant to the Australian Eggs, and we were granted, obviously, the money to work on this particular disease, come up with strain identification techniques so that we can actually trace the source of the viruses that were causing disease in Australia. So all of these, in fact, were happening in vaccinated as well as non-vaccinated flocks. So Australia's had traditionally 2 vaccines that were developed, in fact, at CSIRO. And so these vaccines were used back then, but sometimes were not totally effective. So anyway, so back then, in fact, we had a bit of luxury because Trevor Baggis, in fact, who was an international figure for ILT, was working in our group as well. And Naomi, we can see her picture here as well. She was sometimes, in fact, showing her results to Trevor and getting some feedback as well. So in this picture, you'll see Naomi is showing her sort of viruses grown on cams of the embryonic eggs to Trevor. And as I said before, it was a great environment, in fact, working with the best in the field as well. So first step, what Naomi did, she actually collected a lot of field isolates from different parts of Australia, including Queensland, New South Wales, South Australia, and also Victoria as well. And then what she also did, she used the limited amount of genomic information available to find genes or genomic regions she could use to actually distinguish between different strains. And to tell you the truth, back then we didn't have a lot of sequence available to us. And so Naomi actually found a number of regions and targeted them with PCR. And then we did what we call restriction fragment length polymorphism, RFLP in acronym, and then generated some obviously RFLP patterns that we could obviously distinguish between some of the strains. But what we found soon was it was difficult to rely on 1 or 2 genes to distinguish between all the strains we had in our hands. In fact, what Naomi ended up doing was, in fact, combining the patterns from multiple genes to come up with a classification system. So as you can see here, for example, she used GE, GG, TK, especially ICP4, ICP4 18.5, and TK, B to TK. And then by chopping up these genomic fragments, when you put them all together, could come up with 5 different classes of ILTV in Australia. In fact, back then it was a luxury to have this technique available to us, and it was a technique that was relatively new back then. Took you about a day or a little bit more to actually complete, and it was giving, in fact, a lot of idea about the strain that we have in Australia as well. So Naomi, the other thing she did was, in fact, she selected representative of each class of the viruses, put them into chickens and looked at how pathogenic they were, and got a bit of idea which ones of them were more pathogenic than the others and how they behave as well, including the vaccine strains. Now, so this paper, in fact, published a long time ago in 2006. Naomi left for maternity leave, and we recruited another intelligent lady, Hayley Blacker, and Hayley continued Naomi's work and getting a lot of samples from all over Australia for typing of ILTVs. And in about 2007, we pulled out some viruses which we hadn't seen before, including a type 6, which were found, in fact, in just one area in Victoria, north central Victoria, around Bendigo. And in fact, I don't think we actually detected that virus anywhere else in the country. And the other thing that we found was type 7. In fact, type 7 was a newly introduced vaccine into the country, which was a MSD vaccine called Seva strain. So we typed those as well. And from 2018, we started getting a very large number of strains that we had never seen again. So type 8 and type 9. And some of these outbreaks, in fact, caused by type 8 and 9 were in the farms, both in New South Wales or Victoria, that had used just recently the Cervar strain or MSD vaccine. So that was quite alarming, obviously. And Naomi, in fact, Hayley sort of did a little bit further work in what we call dendrogram and realized, in fact, these type 9 and 8, in fact, were related to type 7, even by doing a very simple dendrogram, which sort of looks at the patterns of this RFLP and try to do some statistical, in fact, equation into it as well. So look, we published a paper, in fact, and we sort of suggested there's a link between class 8 and 9 and the MSD vaccine that was introduced to the country. And the paper, we obviously we got it published in AVJ. And I mean, those of you who publish papers in area of veterinary science, you know, AVJ, in fact, is not the highest impact journal. Nevertheless, this particular paper is one of the highest cited papers I've ever published. And I'll tell you why. So as soon as we published the paper, we got some interesting emails sent to Hayley and also myself, in fact, criticizing that we didn't have enough data to draw that conclusion. In fact, the people who were complaining about these findings were the ones who were behind registration of the MSD vaccine, which makes sense anyway. So, in fact, what it did, in fact, made us to go back and reanalyze our data. And in fact, what happened was we found there's even stronger evidence in there that there's a link between type 8 and 9 and the vaccine that had been introduced into the country. So, I mean, we still kept getting some emails from people asking the same questions or raising the same concern, and couldn't do anything, obviously. Until we had this fellow working with Joe Devlin. So Sang-Won was one of our former students who worked on mycoplasma as part of his PhD, but he came back to our group, started working on infectious lyngotracheitis. So one of the things that Sang-Won did was, in fact, sequence the entire genome of type 8 and type 9 viruses. In fact, it was a very clear evidence that both type 8 and 9 were in fact recombinant viruses that emerged as a result of recombination between our historical vaccines, which were type 1 viruses, and the newly introduced vaccine virus, which was the Cervar strain introduced through the MSD vaccine. So in fact, that sealed off the story we were saying before, in fact, confirmed it, if you wish. And the results, in fact, were published in Science. This is the highest impact journal I've ever published papers, and which was very good for the group. What happens, Sang-Won did a fair bit of work afterwards by putting the type 8 and 9 viruses into chickens, and what he found was these viruses, in fact, are a lot nastier than the other viruses and are more virulent, causing more clinical signs, causing more lesions, and can replicate much faster. And also they transmit a lot faster than other viruses. In fact, he's published a few papers. This is only one of them I've included here. We kept receiving samples from all over Australia, and for a long period of time, in fact, we were getting a fair bit of revenue, which was basically giving us a bit of freedom to do a little bit more work on these viruses. And Rebecca was still working with us. She, in fact, pulled out another virus, which was again different to the viruses we had taught before. And Paola Vaz sequenced the virus and analyzed it, and both of them, in fact, are the first joint co-author of the paper we published as well. And so type 10, in fact, was another recombinant virus that was found in Australia, but this was a little bit different because it only appeared in 2013 and 2014, only in New South Wales, and for whatever reason, it just disappeared afterwards. And since then, I haven't seen that virus anymore. And we still get specimens sent to us from New South Wales, but for some reason, we can't see that anymore. So the backbone of the virus, as far as I understood from Paolo's work, was it was from one type 7 to an 8. So I think what is happening, there's a soup of recombinant viruses, and they recombine with each other as well. And every now and often we get more viruses. And there's some indication, in fact, we have a lot more recombinant viruses around. Now, a few years ago, we had some viruses submitted to us from New South Wales, and according to the technique we use, they were typed as the Cervus strain. So this is the vaccine strain that was obviously introduced into Australia. However, the report from the field was saying that this particular virus, in fact, kills a lot of birds. Mortality is up to 10%, sometimes higher. And it's very nasty, very rapidly spreads through the flock. And based on that, the industry, in fact, paid us a bit of money to sequence the virus, and lo and behold, this virus turned out to be a new virus as well. I mean, still the backbone of the virus was 7, and that's why it was typed based on our RFLP system as type 7. But we named it as type 7B because it's not exactly as the vaccine strain. So if you look at the image I've got here, so type 7 had some very unique regions which we hadn't seen before. I don't even know what other viruses that comes from. We've sequenced quite a few ILTV strains in Australia. We've never had anything looking like that before. There were some areas of the genome which had very high similarities to type 1, 9, and 10. And there were also some other parts that were, in fact, unique to type 10 and also type 7B as well. So type 10, in fact, wasn't dead at all. It looked like still probably is around in New South Wales and perhaps contributes to the new recombinant viruses that emerge in different places as well. So Jawad, who was one of our PhD students and finished, in fact, recently published a paper on his findings, and also that was one of the chapters he, in fact, included in his thesis as well. Okay, so what I reckon is since 2007, the recombinant viruses, in fact, have taken over, and the predominant type of ILTVs we have in Australia and causing massive outbreaks. And I've got a picture of a cane toad here, which is a recent picture showing you how fast they've grown and how big they are and how mobile they are these days. And I think there's a bit of, in fact, analogy, if you like, between the way we've sort of evolved ILTVs in Australia and also the way cane toad has spread and evolved in Australia as well. So the new viruses, in fact, we have, as I said before, have a significantly higher virulence and replicate faster, and also transmit quite fast as well. So yeah, so even as in the last month or so, we've had so many outbreaks in Victoria, and I've been, in fact, invited to a forum tomorrow to talk about these new outbreaks and to see whether there's any, in fact, solutions the researchers and obviously government agencies or field veterinarians to come up with to tackle this big issue at the moment. Okay, so I might actually just move to the next slide. So what has been the a consequence of these typing techniques at APCA, first of all, the industry has learned to be a little bit more prudent with the way they sort of use the service strain. At least they're even more prudent with the use of the way they vaccinate the birds and make sure that every bird in a flock gets vaccinated. And they even, in fact, they're prudent with the use of our existing or historical SA2 and A20 vaccine as well. And the ABPMA, which is a government agency, has in fact renewed or reviewed, if you like, the rules and regulations around registration of new, especially DNA virus vaccines, live vaccines. I know Glenn and perhaps Joe were involved in some forums after the science paper, in fact, was published. They attended some of these meetings to basically assist the ABPMA with new regulations. And also public awareness. These days, it's easy to go to go to forums and people talk about class 1, 2, 3, and 4. And it's actually pleasing to hear a lot of veterinarians, field veterinarians are aware of what's happening around as well. For the faculty and for the university, if you like, there's been a lot of publicities every time the paper published, especially after the science paper. I know there were quite a few sort of media appearances by Joe, maybe Glenn, and I actually got one of my former students from Iran sent me a scan of a local newspaper, as you can see here, and by email. Then it took me a while to find out what is he talking about, because he didn't mention why he sent me that. Just said congratulations. And so I'm reading through this particular article, which is about Barack Obama, if you like, and then read a little bit more, and especially with my Farsi these days, and just not really up to scratch. Then I realized that on the top, there's a, in fact, article about ILTV vaccines can recombine and cause outbreaks in Australia. So the news actually goes quite far away as well. Now, I'm just looking at some of the papers we've published, and these papers have used the strains we have isolated from field in the last 16, 17 years. And just the back of the envelope calculations, I found about 30 papers we've published since then that use these materials we've generated through doing strain ID for ILTV, in fact, at APCA. The grants that I have been involved in and use these materials, if you just sum up the value of these grants are close to $3 million. And I'm sure there are more grants that have used these viral isolates that maybe I wasn't involved in. And I know at least there are 5 students that they, in fact, did their PhD and used these materials in their PhD. So you can see the value of these strain ideas just beyond the value for the industry and or publishing papers. It's about basically bringing more money into the group and also setting foundation for basic research as well. Now, what does it sort of have for field veterinarians? Look, ILT is one of the few diseases that you can, in fact, apply vaccines even in the face of an outbreak. So it doesn't, let's say, spread through a flock of birds that quickly. And there are only 2 diseases I can think behave the same way. And for both diseases, in fact, we can vaccinate them with a live vaccine in the face of outbreak. So the only trouble is the vaccine strains we use for ILT, in fact, cause a bit of mild reaction as well. So, and quite often what happens in a poultry farm, sometimes they don't vaccinate, but as soon as a neighboring poultry farm, let's say a kilometer away, has an outbreak due to a field virus, then they start vaccinating the birds. And then about a week later, they may see a bit of reaction, but it's difficult to judge whether that's a reaction to the vaccine they used a week ago, or this is a start of a massive outbreak. So, and that's where strain ID actually helps a lot. And I've been into positions like this. In fact, this photos comes from a field visit I had in Victoria. Went in there, I see some birds with a bit of conjunctivitis, open mouth breathing. Is it a vaccine that they used recently? Or this is a field outbreak? So some of the birds, in fact, had hemorrhagic tracheitis. So that sort of made me think this is probably not a vaccine strain. So I collected some materials, brought it to the lab. Next day, realized this is not a vaccine strain. This is actually type 9. So this fellow is going to get hit really hard with this virus. So immediately started vaccinating the birds, and it actually worked like magic. So it actually works like water on fire, if you like. So in a sense, the strain ID helps clinicians, in fact, to tackle the disease straight away and prevent the spread of the virus to other farms. Now, so I just want to just deviate from ILTV to other diseases as well. But just before I do that, so the strain ID techniques we've been using for ILTV, in fact, gives us a fair bit of idea, in fact, about the epidemiology of the disease, improves government policies, and generates publicity for Melbourne University and foundation for future research, and also facilitates, obviously, strategies for control in the field as well. So just leaving ILT for the time being, and I want to talk about infectious bronchitis virus. It's a coronavirus, behaves in many aspects similar to COVID-19 agent, which is SARS-CoV-2 virus. The only difference is the IB has been around and people know about it for the last 80 years. COVID is obviously only 2.5 years that we know about it. Anyway, so we started working on IBV in the group it's about 18 years ago, if I remember correctly. And Karim Mardani started as a PhD student with us back then. Karim also works for us right now, in fact, works with us as a research fellow on similar subjects, again, infectious bronchitis virus. And so what Karim did, in fact, he developed a new typing technique based on the untranslated region towards the end of the viral genome. And it's sort of similar fashion as I mentioned about ILTV. So he set up a PCR RFLP technique, and so it was chopping the PCR products with different enzymes and came up with different patterns. And the typing he developed at the end was quite useful, again, to tackle the issues we had in the field. And what happened was Karim's, in fact, project was continued by this smart lady that I've got a photo of here, Kylie Hewson. And Kylie, in fact, further improved the technique with high resolution melt kind of analysis. So if you wonder what it is, in fact, it's basically PCR and with a bit of intercalating dye, put it in this $60,000 machine, and in a mysterious way, the machine after PCR melts the DNA, and when the DNA is melting, then the dye is released, and on the screen you see the basically the curves generated from the release of the dye. So, I mean, the technology is not new right now. In fact, maybe 15 years ago was novel, but we actually developed quite a few strain ID techniques based on this very simple technology. The machine, in fact, wasn't cheap, but the reagents are pretty inexpensive, and we can do it in a very rapid fashion. In fact, in 3 to 4 hours, we could have from the time we get your diagnostic samples, we could have a result for strain ID. So what happened was both Karim and Kylie realized that we're getting, in fact, some odd results with these vaccine strains that we have in Australia. So all vaccines, in fact, we've had in Australia, the live vaccines, are the same serotype, so they should behave the same. And Karim sequenced the 3' UTR of these viruses as well. They looked similar. However, Karim, in fact, so one of these vaccines, which is VIC-S over here, generating 2 PCR bands. I mean, we didn't pay much attention back then, but when Kylie started working on HRM curve analysis, she in fact realized the VIC-S strain in fact generates 2 peaks. One of them lines up nicely with the other vaccines we had, which was I and S strain. But there was another peak that we didn't know what it was. So to cut a very long story short, Kylie, in fact, went back to traditional PCR, sequenced those PCR products, and then realized, in fact, they're different. So the conclusion we made was, in fact, we're looking at a vaccine that has 2 viral subpopulations. And in fact, we were pondering whether that's the reason behind VIC-S being a little bit more pathogenic in the field as well. So if you talk to the poultry veterinarians that are using the vaccine in the field, they're always telling us don't use VIC-S as the first vaccine because it actually causes a reaction and causes a bit of respiratory disease, whilst the other 2 don't. All right, so how do we test that? We need to separate these 2 subpopulations from each other. And I can tell you, I don't think anyone than Kylie could do it. So she did a fair bit of dilution of the vaccine strain and then a lot of cloning so that at the end she had 2 pure cultures of these 2 subpopulations in the vaccine separated, which was a great work. Then she could put them into chickens. And this is actually one of the graphs out of the paper that Kylie published. And what we found was, in fact, the 2 subpopulations generated less lesions and also clinical signs compared to the vaccine strain, as you can see here. This is straight out of the bottle, or the one Kylie made herself by mixing these 2 subpopulations together. So in a sense, separately, these subpopulations were not that pathogenic, and in fact, they were behaving very similar to the other vaccines. But when you mix them together, they have some synergetic activities. If you ask me why this happens, even to this date, I don't know the reason. We've had some, obviously, hypotheses, never managed actually to put them into test anyway. Well, the other thing with this PCR, RFLP, or HRM tests in our hand, we could, in fact, test a lot of materials coming through the lab. In fact, we were getting a lot of samples from New South Wales at some stage, with birds having renal lesions similar to the ones I've shown in the left-hand side. So very severely sort of swollen and white sort of kidneys. And people who have worked with IBV in Australia, they know a lot of the strains we have in Australia are nephropathogenic. And sometimes they don't cause any respiratory disease, they cause just renal disease. And it is very tricky, in fact, to distinguish some of these lesions from, let's say, water deprivations and some other viruses that have been found overseas to cause very similar lesions. I mean, I often do histopathology just to make sure it's not management-related issues like water deprivation. And these cases, in fact, we were getting from New South Wales, all of them had footprints of what we expect from infectious bronchitis virus. So they had what we call interstitial nephritis. So Kylie was testing these the kidney tissues, and all she was getting was the vaccine virus out of these. And surely all these birds have been vaccinated with the live vaccine, so it's expectable to find out, in fact, the IBV vaccine in there. And then we thought, is it another virus, perhaps, because it has had the footprints of an infectious process. Is it something we call avian nephritis virus that we've never seen in Australia? In fact, we developed some PCRs for the tests, and for this particular virus, lo and behold, it was avian nephritis virus. So in a sense, this particular strain ID technique that we use for IBV helped us to, in fact, detect an emerging disease in Australia. Look, I don't think it was emerging. I think it was there before, but we hadn't detected it before. Nobody actually looked for it. But as a consequence, our group was awarded a fair bit of funds, in fact, to look into the prevalence of ANVs in Australia and the damage they cause and the risk factors and all of those. And we got a PhD student, Anthony Chamings, working in this project as well. I need to actually acknowledge Jagota Ignjatovic, and who actually received these grants, and also Anthony Chamings. You can see his photo here, who actually worked on this particular project as his PhD subject. And Anthony obviously set up some strain ID techniques for ANV as well, which was, in fact, very interesting science for us as well. Now, ANV, in fact, is a very, very difficult virus to grow in vitro. So that's one of the difficulties I think the industry had, or the researchers had, to, in fact, detect the virus in Australia. Now, so that was about, obviously, ILTV and IBV and ANV. We've had similar tests for Mycoplasma synoviae, and for Mycoplasma synoviae, because we have a vaccine strain, MSH, so the group has been, in fact, trying to develop tests to distinguish between the vaccine and non-vaccine strains. So there's quite a few tests we've done so far, and the same thing we've done for Mycoplasma gallisepticum. So in fact, what it does, the tests that we have developed for MSH and also TS11 have helped the industry, or in particular bioproperties, to actually market their vaccines a lot better as well. Now, I just want to touch on also the value of the strain ID test we do, in fact, for the our international partners. Nadika Wabagama, she got a grant from Australian government to, in fact, develop some capacity in Gajah Mada University to sort of do diagnosis and strain ID techniques the same fashion we do in Australia. In fact, we've been involved in running some workshops for them and detecting some of the viruses that never detected in there before, including fowl adenoviruses, in fact, in a matter of a day. And we've done the same thing for Mycoplasma synoviae and gallisepticum for Japan, South Africa, Iran, and some of them obviously have generated papers for us as well. So in a sense, in fact, it assists the ID protocols we have, in fact, assists international partners as well. Now, it's not all good news. Developing strain ID technique could be challenging. Each of the tests we offer to the industry probably taken, not a lie to say, 3 to 5 years. So it takes a lot of time, and it needs a lot of, in fact, skills in interpreting results in relation to the field history, clinical science, pathology, and also you should be aware of the limitations of the tests you do as well. And you've got to be aware of the mixed strain infections as well. So you've got to be careful, especially if the birds have been vaccinated before. I was going to actually tell you a little bit about the other things you need to keep in mind and the challenges we have, especially with the interpreting the results, especially with typing techniques overseas. But I think I'm going to skip those a little bit sort of here, because I've got a few more things to show you as well. And same story with ILTV. People have developed ILTV strain ID techniques overseas. However, we're not sort of able to communicate our results that well, because we're using different enzymes, although we're using PCR, FLP, or we're using different targets. And the same is story for Mycoplasma synoviae and so on. So challenges that I mentioned before, Lack of universally accepted targets for pathogens, variation of the target sequences we use, and depending on the fauna of the regions using the strain ID, some tests may not be necessarily useful. So you need to really adapt it to the country or the region you want to use the test for. And some of these, in fact, tests we do, they have no relevance to the, in fact, virulence of the organisms we detect, not all of them. And I mean, you need some equipment to run these tests as well. All right, so what is the future? In fact, this is the slide I used to use, let's say 2 or 3 years ago. I don't think this is future. It's actually current. Nowadays, we've been shifting to whole genome sequencing directly from clinical samples. And I need to, in fact, give the credit fully to this man, Mark Miranda. Is the one who's we've been working together, in fact, for the last 18 years. He's to my point of view, internationally recognized in the way he does things, and it's been in fact, joy working with him. So we've trained so many students together, and one of the things, in fact, has been keeping Mark busy lately is just using this MinION nanopore technology. Basically uses a flow cell that you can prepare your library and just put it on the flow cell and get some fantastic sequencing results straight out of these materials. So we had a student who's just basically writing up her thesis right now, and she developed techniques to use clinical materials directly without trying to grow anything out of it, and get whole genome sequencing of fowl adenoviruses out of them. So this is something that I couldn't imagine, let's say 5 years ago, we could do that. Again, this project goes back to 4 years ago. In fact, we started working on this project with Kinza about 4 years ago. This is before COVID started. So, so I'm just telling you, we have learned a lot about whole genome sequencing straight out of clinical samples, but this particular project started well before that. And Kinza has used the same technique to do whole genome sequencing out of the clinical samples for avian hepatitis E virus. Very briefly, Kinza has also been able to do whole genome sequencing straight out of the clinical samples that come through for pox virus infections. And some of these lesions, in fact, she's been managing to get whole genome sequences are tiny, like maybe even a few millimeters in length. And nevertheless, she can get good sequence out of these, which is remarkable to my point of view. We've had some goes as to try sequence whole genome of ILTVs straight out of the tracheal clinical samples. Results are a little bit patchy at this stage, but we're hoping to improve the techniques hopefully in future. Again, on back of what Kinza did, Martins applied for a grant to Poultry Hubs and won a grant earlier this year, and he's been, in fact, trying to look at the prevalence of avian hepatitis E virus in Australia and type the viruses and set up a strain identification test for the Australian poultry industry as well. Again, this goes back to what I was saying, the strain ID techniques, in fact, gives us capacity to do more testing and get more grants and so on. In fact, this is some images, printed screens I got from Mark just before the meeting. As a matter of fact, you could, in fact, get reasonable whole genome sequence even 20 minutes after you load the flow cell with your products. I mean, this is not, in fact, 20 minutes, but this is about 16 hours, which is overnight. And then we get a fair bit of sequence, in fact, that Mark could get some joy out of it as well. Anyway, so whole genome sequencing is great because it's fast. It's becoming relatively inexpensive these days. Doesn't sort of require isolation or propagation of the organism, which is pretty good for avian hepatitis virus and also for avian nephritis virus. You don't have to do, in fact, anything more. Problem with in vitro passage is it may actually introduce some variation into the organism as well. And it gives you full information about the organism as well. And also has the high capacity, in fact, to detect mixed infections as well. But I need to tell you, there's a bottleneck, which is basically expertise required to analyze the sequences. This is my last slide. So what I think we should do and what I'm aiming for, in fact, we got to sort of expand on what we've found from Kinz's project. We really need to look at various tissues and pathogens, and hopefully we can set up some non-invasive sampling that we could use for whole genome sequencing, which is basically swabbing techniques. And again, that's where information about COVID would hopefully help us in that respect as well. What I really like to see in our group, at least, set up is a platform for whole genome sequencing for infectious agents for animals, and maybe even next-gen sequencing for metagenomics. And that would be useful for production animals, companion animals, and even for wildlife animal diseases. Now, this is my last slide, John. The few people who I didn't acknowledge during my presentation, Polop has been behind a lot of projects we do, especially those that require animal experiments. Also, he supervises some of our students. Denise O'Rourke, I need to acknowledge her because I don't know what we could do without her. In fact, a lot of my students were trained by her in the lab in terms of virological methods and techniques that they learned. Jo Devlin, she's been leading the ILTV projects in our group. Alistair, this young man, I have a lot of hope for this fellow. Alistair's been helping a lot of our students with whole genome sequencing as well. I don't think I need to acknowledge Glenn. Everybody knows Glenn is behind the scene and a lot of projects, in fact, we're on. A lot of you don't know Peter Scott, but Peter Scott is a field veterinarian and he's also an honorary associate professor, and he's one of the links we have with the, in fact, poultry industry. So I need to sort of acknowledge the amount of work, in fact, he does behind the scene for the group. And also Bioproperties always put some sort of effort and money and time of their staff into our projects as well. And with this, I'd like to finish my presentation. I'll leave it to you, John, and I apologize if I went over time a bit.
Thanks, Amir. You might want to stop screen sharing and we'll take some questions. Now, if anybody would like to ask questions, please put them in the Q&A and not in the chat. And while you're thinking of a few questions, let me just start with asking one. And so, Amir, you know, poultry is an increasing part of everybody's diet, and the poultry industry is worth a huge amount of money. Would it be true to say that viruses are the biggest threat to the poultry industry?
With the way we, in fact, raise poultry, especially broilers, yes, that's correct. I mean, the number of kilograms of chickens we raise in some of these poultry houses is basically mind-blowing. I mean, they obviously do it in a welfare-friendly manner. But yes, with this degree of interaction between birds within a very confined space, I think you're right. There's a better chance for the viruses to spread and also evolve.
Yeah, and so I'm aware of the work on Marek's disease, which is a bit similar in some respects to the work on the infectious langiotracheitis virus. So what do you think is the future of the success of these vaccines? I mean, they're recombining in the case of the ILTV, and you've got new strains arising, and in the story you told us they were more pathogenic. And then in the case of Marek's, there was a Marek's vaccine, and then that drove the development of a more virulent Marek's disease virus, and then another vaccine, and that drove a even more virulent Marek's disease virus. And And so there's so it goes on. I mean, are we just in a constant war with these viruses in the poultry industry?
I think we are at some stage. Although having said that, Marek's disease, in fact, is one of the success stories of the poultry industry, because at some stage it was behaving like ILTV, and every few years after introduction of the a new vaccine, we're getting more virulent viruses popping up and different sectors of the industry. But with the selection of more resistant strains of chickens, in fact, Marek's disease, to my point of view, is a disease of the past. And I'm hoping the same story will happen with some other viruses we're dealing with.
Okay, great answer. Let's go to the questions that are in the chat and let's see who's got one. Karim Madani, would you like to ask your question? If not, I'll read it out. Karim, are you there?
Yep. Thank you very much, Amir. Very nice presentation. I really enjoyed it. My question is, are there any differences or similarities between Australian and other countries' ILTVs in terms of recombinations occur?
A good question, Karim. Yes, the recombinant viruses, in fact, have been found all around the world, so it's not really unique to Australia. I think, however, we were, in fact, in a fortunate position to be able to distinguish between these isolates, because for a very long period of time, because of the geographical isolation, we didn't have any introduction of viruses into Australia. And then that's why we could, in fact, go back and check the background viruses we had in Australia. As soon as we introduced one, we could actually find the evidence of recombinant viruses straight away. But if we were running same as the rest of the world with a soup of viruses coming from everywhere, I think the task would have been much more difficult. But I agree, the ILTVs, before the introduction of the Cerevisiae strains in Australia, were pretty unique to Australia. And I mean, there's some similarities between the viruses found in Australia and overseas, but the type of viruses, at least we had, and those top 5 classes I mentioned, they were rather unique to Australia.
Thank you.
Okay, thank you for that. Robin Hall, would you like to ask your question? A bit of a technical one, but let's obviously direct sequencing is fairly new in some of these technologies. Anyway, Robin, what's your question?
Thanks very much. It was a really wonderful talk. So yeah, a bit technical. With the direct sequencing, how many samples per flow cell are you able to run? Like, obviously that's a limitation in terms of cost per sample and throughput. Yeah.
Yep. So you asking a question that I need to refer to some other people, but I can tell you, depending on the type number of barcodes you can include, but I think up to 5 or 6 we've had it in the past, I can tell you. But I need to actually refer it to Mark Miranda or maybe Alistair that can tell you exactly how many barcode we can do per flow cell.
Can we take that offline and send you an answer, Robin, and maybe a few more details?
No problem.
All right, lovely. Thank you for the question. Kevin White here. You're asking a question about ILTV. Would you like to ask it, Kevin?
I think Amir's already answered it. It was the same as the first question about recombinant vaccine strains in other countries.
Right. Okay. Yes. All right. Okay. All right. Thank you. Okay. The next question. I'm just going to read the last question just so that we finish on time, and it comes about the ILT vaccines again. And what is the future of ILTV vaccines? And since recombination is a problem, clearly.
Yeah, I think it's a multi-million-dollar question. So some of you may be aware, in fact, Joe Devlin is leading the project on a gene-deleted vaccine, and I think we have a lot of hope. I've worked with that vaccine as well alongside Joe, and I can tell you it's an excellent vaccine, and it's undergoing registration for limited use, at least in some in Australia. And I have a feeling that it may be the way to do in future. In other countries, people use recombinant vaccines, so basically adenoviruses or Marek's virus, in fact, expressing some parts of the ILTV, and they have advantages and disadvantages as well.
Okay, good. Well, given the time, it's just gone 1 o'clock. I'd like to thank Amir for his really interesting talk. And I'd like to thank you all for taking part today by attending. And I hope you also found it interesting. And I hope you'll be able to join me for the last Dean's Research Seminar of the year, which will be on the 7th of December. And that is going to be on converting genomic discoveries into genetic solutions, but not on viruses of poultry now, but better pastures for the Australian dairy industry. So this is about improving pasture grasses and grazing for the Australian dairy industry, and it'll be presented by Professor Kevin Smith. So I look forward to seeing you all on the 7th of December. Thank you.